After the discovery of penicillin in 1928 by Alexander Fleming, antibiotics have changed the trajectory of modern healthcare, saving millions of people in the process. Despite this, a challenge has risen from the rampant use of antibiotics in healthcare settings, with this being the accidental creation of antibiotic-resistant bacteria. Using increasingly strong antibiotics can combat this, sometimes at the expense of the patient, but the use of phage therapy is a worthy alternative. Recently, there's been a renewed attention about this practice due to its qualities as an alternative to antibiotics. There's also a sense of urgency in this, since the prolonged use of antibiotics result in the decrease in their overall effectiveness. The effectiveness of phage therapy arises from the inherent qualities that bacteriophages have. These are viruses that infect bacteria. As you may know, viruses do not have sexual reproduction or replicative machinery like mammalian or plant cells. They rather have to multiply by hijacking the machinery and metabolism of the host to produce multiple copies. Bacteriophages accomplish this in two different life cycles, known as the lytic and lysogenic cycle. A lytic phage will actively degrade the bacterial cell wall in order to incorporate its DNA into the bacteria. After this, the host DNA has been destroyed, so any replicated machinery in the host cell will replicate the viral DNA, which in turn leads to the destruction of the bacteria when the phage progeny bursts from the cell. There has been major research on specific proteins called lysins. These proteins degrade the host cell wall during the lytic cycle. Knowledge about these proteins is specifically useful for phage therapy, since the goal of phage therapy is to destroy a bacterial infection. If whole phages were used, it could introduce a viral infection as well. This is why using the lysins are important in phage therapy, as they could just degrade the bacterial infection. Specifically, there are two different types of lysins, lysin A and lysin B. While every phage has a lysin A, not all have lysin B. From the literature, lysin B-containing phage are effective in degrading the cell wall of certain bacteria that lysin A-only phages cannot. In this investigation, we propose a novel means to quickly identify and characterize individual phage genomes and groups of synonymal phages, known as clusters, by the existence of lysin B in their genome. By using NCBI protein blast, a known lysin B encoding gene from phage Maru was blasted to find genes from other phages with similar amino acid sequence. These top hits were then checked in the PhagesDB database to determine the phages cluster and subcluster. From this investigation, a number of clusters and subclusters were found to contain phages that also contain a lysin B. Since clusters and subclusters are typically grouped by sequence morphology and similarity, we can assume that if one phage in those clusters contained a lysin B, it was likely that some, if not all, of the phages in the rest of the cluster would also contain lysin B. We then use Centeni, or Famirator, to check the number of lysin B occurrences in the phage in each cluster. This is done by selecting all the phage in the cluster and then searching for lysin B in the FAM map. Draft genes were not considered as part of the total count. This method, while simple, yielded what we expected. We found that our initially selected clusters of A, C, K1, P, and G, that the majority of phage contain lysin B, proving this method to be an effective tool to characterize phage genome per function. Specifically, we found lysin B occurrence rates to be greater than or equal to 50% in all the analyzed clusters. We also did not consider draft genes again. In addition to this, we found that the genes encoding both types of lysins were generally located very close to one another, with a Holland gene downstream. This morphology was conserved in all the analyzed phage genomes, indicating that these genes are likely forming some sort of network motif with one another in order to correctly go through and switch from the lytic and lysogenic cycles. So, in conclusion, the gene encoding lysin B is usually located directly after the gene encoding lysin A. We can also see that the Holland encoding gene is located downstream from this. We also found that our method typically yield a 50% or greater lysin B occurrence rate in the phages we checked, and then we also found that the lysin B gene is typically found in the middle of the phage genome. In the future, we hope to maybe take a look and use our method on more phage. We would also want to do a refined structural analysis of lysin B protein using ITASR and a few other bioinformatics programs. We also want to identify unique gene functions within clusters that contain lysin B, so this would be a more in-depth analysis at the network motifs that arise and that could switch between the lytic and lysogenic cycles. We could also view our research as a stepping stone for a wet lab investigation, so researchers could use our techniques to pull clusters and subclusters that contain lysin B very quickly, and then could use those specific phages in any sort of research as they see fit. Programmers could also use our methods as a structural procedure for a new bioinformatics program that could quickly pull information from NCBI, PBLAST, and Centeni to check gene occurrence rates in specific clusters.